Hi, microbiology students. This will be our first video lecture. You should treat this the same way that you would treat a face-to-face lecture and be taking notes. That's important because it is a proven way to help you remember material. It's a way to document the material you've gone over so that you have a way to review. It's a way for you to mark questions you might have about the material or comments that you want to make. The advantage with a video lecture is that you can actually pause it at any time. That gives you time to write down any questions you have. Also gives you the opportunity to rewind and view things as many times as you need, which is something we cannot do when we're face to face. This first chapter is just an introduction to the types of things we're going to cover this semester, and also a little bit about history of microbiology. In this image, we see a vet that's cleaning up a sea turtle covered in oil, which on first glance may not look like it's related to microbiology, but we have microorganisms that are able to actually eat oil. And as it says here, they are naturally occurring. So they can eat the oil that's causing problems for the wildlife, and when that oil is gone, then they don't have any more food there, and those organisms aren't going to spread and cause any harm. Here we have an image of a lumbar puncture, very important for central nervous system diseases. Take a sample of cerebrospinal fluid, and it should be clear. And if it's not clear, then that indicates that there is infection. Here we have yeast. It says a microscopic view, but that is an electron microscope. The images we're going to be looking at throughout this semester will be through a light microscope, so they won't be quite so highly magnified. And this, when yeast are fermenting, creating ethanol and carbon dioxide, it's what we use for bread baking. This is the Great Sewer, the Cloaca Maxima in ancient Rome. So that was a waterway, then latrines. It was a way to keep the waste from getting in people's houses. We'll see in other slides throughout the semester how wastewater treatment is really important for public health. You don't have to memorize the names of famous microbiologists and scientific thinkers, but we have Western medicine believe that diseases caused by natural and not supernatural causes. Observed survivors of the plague were immune to infection, so that is what led to modern immunizations. And there are great thinkers deep in the past, before we even understood what microbes were, that have led to our modern understanding of microbiology and disease. Louis Pasteur. Lots of inventions. Here I believe he's looking at spinal cords taken from rabbits. That was used for developing the rabies vaccine. And Dr. Koch developed Koch's postulates. Was the first person to directly demonstrate that microbes actually cause disease. Sorry for the abrupt transition. My dog was barking and I had to go take care of that. We're going to be looking at petri dishes, which are circular containers with agar. That is a type of carbohydrate that is a semi-solid media and provides nutrition for our microbes. This has been streaked using this tool. It's an inoculating needle. And this happens to be the organism that causes Legionnaire's disease, but we will be viewing microbes from all sorts of different disease potentials in the images that we're going to see in lab. Lots of different famous microbiologists that there are links there that you can investigate. We had Carolus Linnaeus was the first who developed our binomial naming system. Sort of a uniform way of classifying all living things. In our tree of life, we at one time thought everything was either a plant or an animal. We now know, of course, that's not true. This one, a little more diverse. We have animals, plants, and protists, and we have much greater diversification now. So first we had plants and animals. Then we had plants and animals and protists, and then we added Monera, which are the bacteria. And now we have the 5 kingdoms: plants, animals, fungi, protists, and the prokaryotes. Now this, by Woese and Fox, is called the domains, which is a broader category than kingdoms. These branches of the trees are based entirely on ribosomal RNA. So we have 3 domains: bacteria, archaea, which means ancient ones, and eukarya. And these are based on the cell structure. Only the eukarya have a nucleus. The archaea and the bacteria have no nucleus. They are living. They are cells. They have DNA, but they do not have a membrane structure around their DNA. This shows us our relative sizes. Frog eggs are quite large, 1 millimeter. You can see those without the light microscope. The light microscope, like the ones that we would use in the lab, and you have a virtual microscope in your lab archives notebook. We would be able to see human eggs, pollen, animal and plant cells, red blood cells. With really great staining, you can see mitochondria. We'll definitely see slides of stained bacteria. But once we get down to most viruses, they are so small that you would not be able to see them with a regular light microscope. You would need an electron microscope to be able to do that. There are different bacterial shapes that we'll be talking about in lab. We're going to be doing identification of an unknown bacterium. And one of the first things we need to know is what shape is it. The shape alone will not identify the microbe for you, but it is one very key characteristic. And a coccus is a perfect sphere. Bacillus, or a rod, look like little Tylenols. The vibrios are curved. Coccobacillus is what it sounds like. It's like if coccus and bacillus squished together, so it's an oval shape. And then we have spirillum that are wiggly, and spirochetes that are really true corkscrews. The organisms that we've used in our face-to-face microlab are the same ones we're going to be using in our virtual one, even though we don't have risks of infections. And our organisms will either be coccus or rods or bacillus or coccobacillus. We don't have any vibrio, spirillum, or spirochete, but there are certainly shapes of bacteria that exist in the world. The archaea, the Archaea, live in extreme environments. This is at Yellowstone National Park, so it is extremely hot, and it is containing fairly caustic chemicals, and those microbes are adapted to live in that environment, and they are hypothesized to be very similar to the microbes that were on early Earth. Diatoms are algae, so they are not bacteria, they're not archaea. They are eukarya. They have a nucleus. And diatoms have cell walls that are made of silica. And silica is a component of glass. So they do look literally like little glass boxes. Giardia lamblia is the main reason why if you go hiking in the backcountry that you bring a water filter. It is very common in the digestive tracts of mammals. And when their feces get into water supplies, that is how we can contract Giardia. Candida albicans is a yeast, and yeast are a fungus. And even though they're really small and can be about the size of a large bacterium, they are eukaryotic. They have a nucleus. And this is shown as the yeast have been budding and are forming clumps. It can cause vaginal yeast infections, it can cause oral thrush, and it can cause skin infections as well. So Candida albicans. And there are fungi that when they grow large enough can cause food spoilage. We're probably all familiar with that, with the leftovers we leave too long in the back of the fridge. This is a tapeworm. It's a beef tapeworm. It says, the one shown here can reach 4 to 10 meters. 1 meter is about 3.3 feet. So they can be many, many feet long. And of course, a beef tapeworm could become quite much larger than a human one, but human tapeworms can be extremely large as well. And we would contract that from eating undercooked infected meat. An image on the right is of a guinea worm, and it's being removed. As the worm exits the lesion on the patient, they wrap it around a stick and pull it out. So as we progress through our disease chapters later in the semester, it's probably a good idea to plan your mealtime around the images. Coronavirus. This is an older slide. This is not the novel SARS strain of coronavirus that causes COVID-19, but it is the structure of a coronavirus. And it's called that because when you view it under an electron microscope, they seem to glow or have a corona around it. On the other side, we have Ebola virus. It's the filovirus, which means filamentous. So we have completely different kinds of shapes that can happen with viruses. So we're looking here, a virologist out in the field looking for influenza because avian influenza is a type of virus that can go from the animal to humans. So there is our phylogenetic tree of life. We've got the eukarya here, the archaea here, and the bacteria over here. And these organisms, the archaea, are the ones that can live in more extreme environments. They are hypothesized to be more like those ancient life on Earth. But what you see from this phylogenetic tree, where there's a branch point, means distance of relatedness. So here's our common ancestor to everything. The bacteria go one way, the archaea and the eukarya go another. And that says that genetically, all of us in the eukarya are more closely related to the cell structure of the archaea than we are to the bacteria, because the common ancestor here is the common ancestor to all of us. Whereas more recently is where the eukarya split off from the archaea. Back to our looking at the types of organisms and cells that we can see with the different types of light microscope. And that wraps up chapter 1. I know I went through it really fast, but I did that because I know you can pause it, and I want to save video space, and you don't need to listen to me ramble at you for an hour, because in class we would be taking the time to stop and ask questions. We don't have that luxury in an online class, but we do have those discussion boards. They are required, so you get points, but it's also your opportunity to ask questions, get your colleagues and fellow students to answer. I will chime in. If people aren't answering your question, I will pop in there and help you out. If your fellow students are providing incorrect information, I'll correct it. But if you guys can help each other out, form sort of online study groups, that is the way we can build community and connection in our online class. So the current deadlines for this video are to post your question by noon or 11:59 a.m. on Monday, August 24th. And then you have until midnight to go back in and reply to your classmates' comments and questions. If you don't reply first, you don't get a chance to reply to your colleagues. So let's try to get off on the right foot. After this week, I will be doing a much better job of getting our videos posted at least a couple days in advance to give you more time to work on it. But we're all easing into a new learning environment. We're going to do our best to stay on track and to be patient, kind to each other, and to do the best we can. If you run into problems, let me know as soon as possible. There's might be little I can do to help, but at least I'll know what's going on. So take care. I'll be like Seth Meyers. Wear a mask, wash your hands, and stay home as much as you can. I'll see you in the next lecture.